Let's see, how do I want to start this? Pollinators. All right, so let's start with the PowerPoint. Just going to show you the pictures of them, the very basic stuff, right? Because you guys can read this and you can use the Quizlet to study. So I would rather spend more of my time working with you guys on the report and or the proposal and the data sheets and your other aspects of the project. Are you guys okay with that?
Yes.
Perfect.
Sounds good.
Okay. All right. Let me share this screen. I'm just gonna head down here. All right, so this will be your third set of pollinators that we have gone through. And again, 10 every week. Now, I will say when you go to take this quiz, you guys are going to have 10 for week 2 and 10 for week 1. But when you go to take this quiz, I'm going to start back adding some species, right? So if we have species that are starting to look like they could almost be the same, you may have 12 or 13 questions on your quiz. So just be aware that you may be now adding in some from the previous PowerPoints, okay? So the first species we have is the red banded hair streak. This is one that I see very commonly down here on the coast. These are 7/8, the 1 1/4 inch long. The main thing is that whenever they're sitting on a leaf and they've got their wings up, which we'll talk about that in a few minutes, what these things are called, you're going to see that bright orange streak and this kind of like bluish gray patch back here. They call that a hindwing blue spot on these guys. And again, these guys are everywhere, but they really love sumac species, wax myrtles, sweet pepper bush, and tall goldenrods. And the goldenrods down here and on the coastal area are everywhere right now. I'm not sure, it's probably very similar in the upstate. They do like a highly varied, though usually prefer to be closer to forest edges and like it to be a little bit more moist. Continuous flight, March to October. They have at least 2, possibly 3 long broods. This is the variegated fritillary. It's going to have more of a dull orange color, very pretty variegation in those wings, which is, kind of what you're seeing here. 1 3/4, 3/4 inch long. See, the underside is going to be more dull, kind of a brownish color. Again, found everywhere, but they love maypops, violets, and clovers. They do prefer those strictly open areas, so I tend to see them more in the pastures. Found March through December, 3 broods, rarely 4. The cabbage white is pretty distinct. Every now and then a little yellow, or even more specifically who else would you see that does this? Probably the cloudless sulfur. Every now and then a cloudless sulfur will get white enough and the markings get faded enough that you might kind of confuse it with this one. But every now and then, but this one is still pretty distinct. Going to be that white, very creamy overall, 1 3/4 to 2 1/4 inch. Found in mustards, cabbage, broccoli, kale. So they basically like anything in the Brassicaceae family. They like those open country habitat, cultivated fields, March through November, 5 or more broods. So you should see these about year round. We have the summer azure. I think that's the one I just caught. I think somebody up there. Nope, that's not the one I caught. That's the one that Ty caught. Okay, Ty caught this guy. Yeah, so male above, medium blue, whitish leading front wing edge. Very small butterfly and very unique markings on the underside because it's not blue at all. Has multiple broods per year from mid-February into April and then May into late September. So you're going to see them in 2 different spreads. They like herbaceous and woody plants, mostly things like dogwoods and shrubby species. The red spotted purple is going to be a beautiful city blue to dark black on the upper side, and just really, really beautiful colors with orange red marks on the bottom, some blue mixed in with that submarginal band of orange. 3 to 4 inches, so a very large species. Host plant going to be black cherry, tulip poplar, and black willow. So you guys should have a good chance of seeing these up there. Typically going to be April to late October, so we're about to lose our chance of seeing them. 3 broods, possibly 2 at higher elevations. We have the hackberry emperor, which is going to be just a very basic buffy brown to kind of sooty black on the exterior of the wings. It's going to have one very obvious large black eye spot on the lower portion of the forewings, which is this part right here. Above the hind wing shows a submarginal row of those large black spots. So you're looking for these spots basically. It's going to be about 1 7/8 to 2.5 inches long. The underside is going to be that more buffy brown, but you're going to see that very distinctive row right here. Be careful because from this side, you may confuse it with a Carolina satyr or satire, however you spell it or pronounce it. 2 broods over most of the region, April to September. This is the American snout. It's got a very distinctive shape to its wing as well as a distinctive snout. So you should not confuse this one with anybody. And again, that wing shape is very distinctive. So as long as you see the snout, the wing shape, and the orange, that's your American snout. Found pretty much everywhere. Has 2 long living broods, so it's seen nearly all year. It prefers hackberry and sugarberry. We have a lot of sugarberry down here. Dirt roads, wide trails, bottomlands. First brood will be like April to May, second brood late summer and fall. The question mark, again, very distinctive wings. 2 seasonal forms, so that's the thing you're going to have to be careful for. You're going to have that mostly bright orange version, and the summer form is going to have those dark brown hind wings, and the winter form is going to be bright orange and might even look almost black towards the tips. It's a 2 1/4 to 3 inch butterfly, so again, very large. Likes hackberries, elms, and nettle. Flight period is pretty much any month of the year, but it has 2 long living broods. The eastern comma, again, another very distinctive species, going to have bright orange above with numerous black spots and bars. The summer form will be brown to black on the hind wing, and the winter form is going to have that bright orange with black spots on that hind wing area, 2 to 2.5 inches. And wingspan and width, 2 forms, again, both are going to be orange above, summer form with black, and winter form orange with an orange hind wing. There's a little bit of spotty absence on this species in South Carolina, but in our area, we should see it in all months, 2 broods in most places. Does prefer to be near moist hardwood forest. So I have not seen this yet, but if you guys pick a good location, you might find this one. All right, told you that one was going to go quick. All right, so the next thing I want to talk about real quick is I need you guys to just go through your data sheet and make sure that this is the way you want it to be. You can add additional data sheets on the back end or change it up. This is really where you guys get to make your first methodology decisions on what information you're going to collect. Now, all of this is information that I collect. So if you collect this information, you can compare it against my data. If we're looking at, say, we're both looking at a prairie, you could compare your prairie data on the dates you surveyed to my prairie data within those same time frames. Somebody is not muted. Hi, can you mute? Oh, lost him. So you can change some of these or you can leave them the same. Honestly, you've got the Kestrel, so collecting these variables is not that hard. Deciding what the clouds look like, it's looking up at the sky, rain totals. Anyway, the more you collect, the more you have to make comparisons against my data within those time frames. The only thing that is really missing here is that I do not have the wing length on here because on my study, I'm looking at monarchs, and I'm actually doing wing length as well as wing condition. You do not have to do that because I do not have the monarch tags to send up there right now. Um, but you can adjust the things that you're collecting here to um, more match my data. You can see here the different type of activities that these animals can be doing. We have roosting, and I took the time to actually put descriptors down here for you guys. This isn't actually on my sheet. I think I want to change it because every time I go out there, I'm always kind of like, okay, let me remember again each one of these means. So you have roosting, which is going to be closed wings tucked away at dawn, dusk, rain, and clouds. You can have nectaring, which means they're going to be actively in that flower, getting the pollen out. Flying, moving in the air. Basking, they're going to have their wings open and they're just going to be sitting on a leaf. If they're ovipositing, the female will be alone, but you'll see her abdomen kind of bending under the edge of the plant or under the edge of the leaf as she's depositing her eggs. Resting will have a single butterfly just sitting quietly, no feeding, not looking for any sort of sun angle on those. Mating, you'll have two butterflies connected at the abdomen, usually opposite facing directions. Ty actually saw a new activity that was not in any of the literature that I've looked at so far, at least as far as like copying it, but it was called, I think he called it puddling. Ty, are you available to speak to explain what that is?
Yes, hi, sorry, I'm at work, but it's pretty slow right now, so I was able to join. What was the question again?
What is puddling? Because that's not one of our descriptors, but it was interesting that you found a new behavior. What was puddling?
Yeah. So puddling is when they like butterflies. Like I think me and I can't remember who went out with me. I think Vicky and Jenna or Jenny, but we went out and the eastern tiger swallowtails were all circling around puddles of water. It's what they do is they like take in the water and they're trying to get the minerals through the water, and so like they're puddling to get. I think Crystal, do you remember what minerals they're getting when they like drink that water when they're puddling?
I did not, but now I want to look it up because that's just data, and I love data.
I
Um,
think it might be sodium. I'm not sure, but I think it's similar to when like some um, butterflies will congregate on like animals and drink their sweat. They're like getting the minerals from the water and when they puddle.
So yeah, and then we can extend on that. They're searching out sodium, nitrogen, and amino acids from that damp soil and the animal waste, which is not typically found in their flower nectar. And that is essential for their reproduction, particularly for males to pass to females the egg for egg development and for overall bodily functions and hydration. So that's pretty cool.
Yeah, it's pretty
Um,
cool. I'd never seen it before.
yeah, I still haven't seen it, but yeah, we're not gonna record that because none of the data is actually has looked at that. I'm not. You can choose to record that. That could be something a variable that you guys look at. Um, so again, you can change up what things you're collecting, and you would just adjust these sheets as needed. And then on the back, one of the things I have no idea what's happened to my things there. It always ends up having a bare back sheet, which isn't going to hurt anything. And I'm not sure where my lines have gone, why they're not showing up. But typically, you're going to do a plant survey, so just have your iNaturalist app out ready, or a herbaceous identification book, and just get out there. And when it's asking about um, this right here, where it's talking about, where did it go? Forage habitat cover, it's specifically talking about the number of flowers. So if you went out into the field and it was like nothing but flowers for as far as the eye could see, and there was hardly anything else there, you'd probably be reaching, that'd definitely be above 50% forage. It is unfortunately a discretionary variable. So you guys just have to make your best judgment on what it is and record based on that. Again, your families are going to be right here. Even if you're not 100% sure, you can start to kind of get it down to what family it may be in based on just some of the common traits by reading these. But again, if you can get a picture of your species, either in the jar or after you've already put it to sleep, you can hold it in your hand gently and take a picture of it. You should be able to ID these things really well. It's very, very important that you always mark down what site you're at, because that habitat is going to be attached to the species, especially as you're putting them in your pollinator collections that you guys get to keep at the end of all this. And again, you're going to collect all the flowers. If a pollinator lands on the flower or the plant to nectar, that would be what that associated plant is. Or if they're resting on it, typically you're looking for pollination. Then, but if they're resting on it, you can write that. I do not try to go down as detailed with grasses. I only try to get plant names with associated plants. The associated plant becomes a number within your data. The plants themselves over here just become a present absent, yes or no, 0, 1, essentially, like binary within your data collection, which that's coming up as soon as we get into that. We'll talk more about that. I can kind of show you real quick what mine looks like. Let me find it. This one. Oh, that's the survey, right? Crystal. There we go. So essentially, can everybody see this? I know when you're on a laptop or if you're, not at a big computer, it can be a little bit difficult. I've got these blown up a little bit. I haven't got all my data put in. I lost my interns, so it's been kind of hard. But basically, all I do is you can see right here, this is all the same day. So you're going to put in the same date for each species. It's always going to be whichever site you choose for that species. For me, I added in a couple of other variables, but there's my rain totals, drought, forage, sky, each one because you do 2 readings. These are the times that they were collected, transect, like how far away were they from the center line of that transects, which you guys will learn in the field, and then the family that they're associated with. Whenever I collect a butterfly, I create a metadata sheet in the back, and I can share this with you guys so you kind of have like something to look at, because I know if you're like me, I'm very visual. I like to have something that represents where I'm going so I understand what I'm doing. But I essentially create all of these like codes. If it's the family, it has a code. Unknown, if I couldn't ID it, 0. Hesperity, 1, 2, 3, 4, 5, 6, 7. For the plant, I put in their full name. Then I put in their abbreviated name, and I try to always keep these things in alphabetical order on the front page. And then I put in their common name. But no matter what I'm doing, I always know what it's referenced. Same thing with pollinators. Find it, the abbreviated name, common name. I've got numbers for droughts, 0, 1, 2, 3, 4. I've got numbers associated with each one of those activities. I feel like I got a comment in the chat. Oh, yeah, yeah. Yeah, he told me it was a lot. I think Ty sent me a picture and I was like, oh my gosh, that is so many butterflies. Sorry, I just saw your comment. The plant association, and this is even when Keegan was trying to help me put data in last week, this was a little confusing for him. So the first time, basically, he had the plant association is the plant that they landed on, but the plant association itself, if they ever utilize one of these plants, it just gets its own number. So as more plants get used and get added to the list, the higher my numbers would go. That way, when I'm in the back and I want to compare, so this is all my abbreviated butterfly species here at the top. A 0 means it was not there, a 1 means it was. I still have to put each species in their own line. So there'll only ever be one number 1 in a single line. Even if you see more than one butterfly, it's still only, yes, will do, Ty, thank you. Even if you see 2 butterflies in the exact same moment, they are going to have different activities, which means each species has to go on its own line, even if it's seen in the same time. There's the activity that they were doing. So, if I go back to my metadata, I can see here that the activity was 3. So that guy was flying. So you want to always make sure you create a second tab metadata sheet. Then these are my pollinator species. Now, my plant. The thing about the plant is once you record those plants will always be present in that day, right? Because flowers aren't there that morning and not that afternoon. They're always present. So the plant will be something you can copy and paste across the entire day that you were doing that survey. If this is very confusing, we'll continue to talk about this to make sure you guys are good. And both Ty and Drew have done this. And again, as I'm adding new species, I tend to right click and hit insert and add the species in alphabetical order, because if this list gets bigger and bigger, it just becomes harder and harder to manage without that. And then here you see that plant association. So if I said that this pollinator was doing a whatever number 2 is, so I can go over here and I can see that at number 2, this guy was nectaring. And then if he was on nectaring, what plant was he nectaring from? I can go back and look at the plant association and say that he was nectaring on white clover. So this is how you're going to build, the easiest way to build data sheets to be able to utilize that data in R. One thing that I did add to my surveys that I had not done before, and you'll probably very rarely use it, is a rain gauge, because I realized I wanted to collect, is it actually raining that day? And if so, is it no, like, is it no rain? Is it a light rain? Is it a medium rain, moderate, or heavy? Again, that's going to be discretionary to the person involved with the situation. But, maybe we will see some pollinators out there in a light rain, but not in a medium or hard. So I'm just trying to collect as much data as I can. You do not have to collect all this. This is your research. All right. Anybody have any questions there? I'm just starting to kind of introduce you to the data sheets, not having you guys do that. You guys do that just quite, unless you start surveying this week, you will, but we got to talk about that next. Okay, so no questions on that. Let me get to the paper real quick. I had it pulled up, but I have so many papers pulled up, it's kind of hard to see. All right. I felt really brutal when I was grading it, and I was trying to be fair, but also give a lot of constructive criticism. So I hope nobody got too mad at me on this. And again, it's not weighted as heavy as it looks, overall. But I want to kind of go through this paper. So who here has already looked at the comment? Oh, I see Jenna. So there are a lot of comments. So I just kind of want to break it down. So I saw you guys had a really great start in this paper, and I know that Drew has already said that you guys are like, whoa, we need to slow down. We're not ready for methodology yet. And even if we don't do methodology, I need you guys to pick some very basic things you're going to do. Like, we know we're going to run a 50-meter transect by 25 meters wide. That's what we know. So we're going to at least start our surveys, because we really need to get out there and start running surveys. And collecting data, even if you haven't completely hammered out your methodology. And I'll be very honest with you, as I've been doing my surveys, I added rain 2 weeks ago. My methodologies will adjust, just like yours may adjust as you kind of move along. So what I did was I kind of looked at your overall paper to see, where were you guys going with this? What objectives were you kind of leading into? And I know we had put some kind of like some guides in there before you started writing. And I restructured those just a little bit. And you can switch this around, but I was trying to find a flow that worked really well. And so what I would like for you guys to do is look at the structure, pollinator and plant interactions, right? Because that's the first thing you guys started talking about. The ecological role in aiding the dispersal of pollen, I want you to basically come down here, and if a sentence fits better into one of these headers, drop it underneath. Let's restructure the paper and then kind of go back and read it and look at where the gaps stand out again. And so and that's what I'm saying, restructure your writing below into these themes, making sure that the sentence fits, has at least one source, but preferably more. And I see there's a lot of things where only one source was used. You can just put that same data in the journal search, and you'll find other articles that will either agree with what you say or even contradict it. And there's nothing wrong with saying in your paper, in this journal or in this research they found this, but in this research they found that, therefore We're going to do our research because we want to see what we find. There's nothing wrong with that. So you may even have some contradictory information in your background, which justifies why you want to look at it a little bit harder. So again, get on these sources a little bit, um, read over some of these comments, and just start addressing them one at a time, going through the list. But I think the first thing to do would be to grab all of these things up and restructure them into sort of these categories. Does anyone here, because I feel like there's I think nobody's really responding except Jenna, but is there anyone here that has concerns about restructuring the stuff that you've already written to kind of fit into these categories? Or do you think there should be a different category, or this is too many categories? What are your thoughts?
No, I completely agree with you. I think we could restructure everything. So yeah, just give us the time to go back and relook at it, and then we'll get it put into place.
Okay.
I agree. I like the categories.
I agree
Okay.
with that as well.
Okay. Yeah. So, I mean, these are the categories that stood out to me when I read your paper. So I was hoping that this would be okay with you. I just, like I said, I think it was just more of a flow issue, and there's a little bit of gap in missing data. And I put the really detailed comments within the rubric within Canvas. So if somebody wants to print it off, you guys can sit down and kind of go back through this and try to figure out, like, okay, what can we add? What can we miss? And kind of fill it out a little bit more. That's really what I'm looking for. I think at that point, if you guys, I know I have communicated with the South Carolina Botanical Garden. They did respond back to me for putting in that research request. Their main concern is that Honeycutt Creek has a lot of sensitive vegetation on it, not the section right there behind the parking lot, but basically from that section all the way down to the other end where it's up there at the cabins. And their concern is if they permit research there, there may be damage done to the trail. And they want to meet, which puts me in a weird situation because I can't get over there. It is possible that if one or two of you are able to go meet with them and I can be on a Zoom call or a Teams call with you guys, that we could at least talk that way. If anybody here is willing to do that, you can put a hand up, react, raise hand, whatever, so I can get your name down. And I can try to schedule a time for you guys to, like I said, at least two of you probably to go out there and talk to them. I think the main thing is, one, addressing whether they actually want us to use that, if we can be careful. And if not, are there other areas within the park that does not have such sensitive vegetation that they would be willing to allow us to use it if it's near water? I know they have ponds down there, but I'm not really sure ponds sitting right in the middle of a flower garden are representative of what we're trying to do here. You'd probably get some interesting information about what pollinator species are visiting the garden, but I'm sure a lot of people already know. So that's what I need to get cleared up is, can we use any place within the botanical gardens? And if we promise to be extra sensitive within Honeycutt Creek, would they still allow us to use that area? So do I have anyone that is willing to meet with them okay, so Jenna's willing to meet with them if I can get a scheduled time. Jenna, has your schedule changed much? Okay, so Vika and Jenna. Okay, have your schedules changed much from whenever you put them into that document? Because if not, I'll just coordinate a time to meet with them that fits that document. If you can give me another thumb up, if that document, the Excel document, still is pretty representative of your availability, I'll use that to try and get you guys set up with them.
So mine hasn't really changed, but it's hard because it kind of varies like week to week. So like I was able to make today's meeting because my dendrology class went to the mountains and wouldn't be back till later, and I couldn't do that since I had to pick up my kids. So
Yes.
like on days like today where I can't go to other classes, then it opens up time, if that makes sense.
Yes, it does. I'm going to try to stick to the times that you and Vika are already open and
Okay.
at the same time, so that I can try to coordinate that with the gardens, and if you guys can get down there. So let me write that in my notebook of my handy dandy notebook of things to do. Coordinate Ika and Jenna to garden. Yeah, and girl, I tell you what, I feel you. I never got to do the mountain trip. There was a lot of things that I didn't get to do when I did dendrology for the same reason. But I
I'm
survived
not mad that I won't be stuck in a van for hours, to be
for
honest.
hours.
Yeah.
And that hike up that mountain is something I've heard a lot of people say they almost didn't make it. But all right, so let's see. Is anyone here struggling with the concept? Well, how about this? Let me go here. Other than talking about the paper and making sure that you guys understand what to do, I don't really have anything else to cover today. I am going to continue recording the lecture if you needed to leave. What I do want to address is anyone who is still feeling uncertain about the writing process, or just, I know sometimes when I go to write a paper, especially when I first started writing them, I really just didn't know how to search for sources and how to put them together. And so if there's anyone that is feeling uncertainty on like where to go or what to do, I am more than willing to talk to you about that. I do need you guys to clean up the objectives just a little bit. And I think I wrote that down here. This isn't really an objective here. Objectives look more like this. It's always like a power word, like an action word. You're going to quantify patterns of species richness, relative abundance, and community composition of Lepidoptera within 3 distinct wetland ecotones. Um, 2, you're going to examine, right? That's your action word, how environmental and habitat characteristics influence patterns of Lepidoptera distribution and abundance among 3 wetland ecotones. And then you guys, as you're looking through this other stuff, come up with your third one. And it could be something you've kind of already written up here. You just need to have it presented in a way that is power word. Here's what we're looking at. Here's why. Does that make sense? Because you don't need as much in the writing part of this in the objectives. As a matter of fact, objectives, finally, the team will also conduct research on the distribution of these in upstate. The team will evaluate the extent in which climate change is responsible in driving the migration of Lepidoptera towards. So this right here, when I read this, we will evaluate the extent to which climate change is responsible in driving the migration of Lepidoptera towards higher latitudes and altitudes where conditions are more favorable. This was interesting to me because this right here, see, in the background, you should be talking about that, because the background is what leads to your objectives. So what you want to do is find papers where people have already started seeing things like this. And if you do not see it in Pollinators at all, you can find no paper whatsoever that talks about behavioral or any type of pattern changes or seasonal changes in pollinators that are potentially related to climatic variables. That's your novel research. If somebody has done it, but it was only in coastal South Carolina, it's still your novel research, because now you're checking to see if you're seeing the same things in upstate South Carolina. So that's how you're creating your objectives. So I really do think that this is important, because that could potentially be a research question. Because down here you say quantify patterns of species richness, relative abundance, and community composition within 3 distinct wetland ecotones. You could also have one that says quantify patterns of da 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 based on latitude, based on all elevation. You can call it whatever word you want. Within wetland ecotones, or within, whatever you want to say. See, how else could you word that? The team will evaluate the extent to which climate change is possible driving. Somebody give me an idea of what they think, what other kind of change do they think climate change could cause to happen in butterflies?
Maybe something to do with the plants they used, the host plants. And it would obviously hurt them.
Yes. Okay. So what could potentially be happening to host plants as temperatures are changing, or what we might we see with host plants?
Their like distributions or ranges change, so where they were once available for butterflies, they're no longer available, or vice versa.
Yes, so we might be seeing some species extirpation or some species creep introduction. We also are starting to see bloom time change. And think about if things are blooming earlier, but the broods aren't coming out. Let's say that for 1,000 years, everything was blooming April 1st, and the butterflies that utilized those plant species were emerging April 1st. It all lined up. If the climate patterns are impacting bloom times, you may now have those same species blooming March the 1st, and the blooms and the resource has already been used up by the time the pollinator emerges April 1st. So when you're thinking about your background, look for things like that. Look for, because now we've got your outline, right? So there's your climate change. Start digging into the literature that is telling you what are we seeing with pollinators, and figure out what we haven't looked into yet. That then translates into that objective, that really strong objective on the back end here. Then those objectives will lead into your methodology. Because if you want to look at climatic variables, which variables, right? So, okay, your methodology is what tools are you using? If you're using, a Garmin, and you're using altitude, an altitude setting on the Garmin, because maybe you're doing a survey at the bottom of one of these mountains, but you decide to drive up and do one at the top. Those are the type of things that you want to decide in your methodology, and then what tools you need to use in order to accomplish that. And especially if you need tools like a Garmin, which I haven't purchased for you guys yet because it's not in my research, I need to know these things so I can get this kind of stuff up to you. So that's the reason that getting on the methodology is so important. I will push the methodology back, but I think what I'm going to do is just go ahead and set the methodology to the week following where I was going to put it in, and have you guys restructure this part, but also start adding in the methodology based on your objectives. Your methodology, so you need to know your 3 sites, because the first part of your methodology is always going to be the study area. Where are you studying? Where are you researching these things at? If anybody has had GIS, you can create a GIS map that highlights the 3 areas. If you haven't and somebody wants to work with me individually through Teams, we can try to do this together. I'm telling you, GIS is not my strong suit, but I can do basic maps, so we can learn those things together. That's the part about this class, and why I'm trying not to be super hard on you. And I promise, even if your grades look tough, like I graded this where I graded it, but I'm usually a curve grader, and as long as I see people are giving an effort, I'm not trying to make it hard, but I do want to give really good feedback and constructive criticism so that you improve. I think I've said way too many times today. Any questions, any concerns on any of that?
I had a question about collecting.
Yes.
How do we know which butterflies we want to collect and which we just want to study and leave alone?
So usually when you first start pollinator collections, unless there's something that is so rare that if you killed it, you may never see another one again, you typically collect them for your personal collections as an entomologist. If you think that this is something that you will enjoy and you may continue doing, and you want to be 10 years from now with a really awesome pollinator collection, because maybe you want to go into education and you want to share, or things like that. And let me close this out for a second. You just keep collecting the pollinators. Now, one of the things that I need to do for me specifically is, as you start to get samples and you collect them and they're pinned well. I'm going to tell you, I've destroyed a lot of samples trying to learn how to pin. But as you get them and you get to a place where you're pinning well and you're building that collection, have yourself a running list, because each one of you are going to have your own collection. So you can determine which ones you want to keep and how big you want your collection to be. And will you ever use these collections again? I would like to see at least all of you have 5 by the end of the semester, 5, you know, decently pinned collections or specimens by the end of the semester. But you can make the decision, and yeah, keep a good running list on the ones that you have pinned and you have pinned successfully, so that if you're out there in the field and you've started, you know, at this point you've captured 30 butterflies and you're like, oh man, that one looks so similar. Have I already got this yet? But you don't want to keep euthanizing the same species over and over again. If every time you go into the field you have a collection of everything you've already collected, you'll know. And that way you're not doing repeats. And I'll show you when I talk about the education side of it. So in the spring, those who stick with me, we're going to start learning about bees. And today, this came in the mail for me. I don't know, again, I know you guys can't see it, but this came from, who was this? US Geological Survey. I'm working with someone from US Geological Survey. And I said, my friend, I do not know how to identify bees. There are so many and they all look so different. So this gentleman, who is pretty much a bee specialist in the United States, says, I've got something for you. He has spent a lifetime collecting these. And he says, I think it will do you more good than it will me because I have learned how to ID these. I'm going to send you my entire collection and you can keep it. And you can use it because I've already got everything labeled. And you can use this to practice and learn your bees with. So, you know, if I was to go into a school and I had a beautiful butterfly collection, I can be like, hey, look at my beautiful butterflies. And here's what they are. And here's what they go after. So your pollinator collections are what you make them. That's the long answer to your very short question.
Thank you.
Okay. Yeah, these are my new specimens that I got Monday for me. So remember I was talking earlier about we have the cloudless sulfur and a clouded sulfur. The cloudless sulfur is this top one. It's going to be larger and it's going to be yellow, and oftentimes sometimes they can even look white, the females, but the clouded is going to be more of a neon green, and it's going to have this black band on the edge of its wings. And when they're flying, the only way you can tell them apart, right, because you got little wings flapping, is going to be that neon green. But even if the neon green isn't there, you can tell based on the fact that you'll see that black border on the clouded. So I just remember black cloud, black clouded sulfur. This is how I'm getting there. And then I got my first orange, so I was really happy about that. So these are my new ones. So I'm trying to get to a place where I'm only collecting them if I need really good samples or I have never collected them before. Okay. Where else? Okay, last thing, and then we're good to go. I know we're working on the garden, which may or may not happen, so we definitely need a backup site. You guys know when you're available. Drew and Ty know how to access the stuff within the lab and can get this to you guys if you're going out on a day without them. But I would say it's probably important to try and go out with them on a day where they're available. Otherwise, you're winging it, and they've done it before. I wanted to try and come up this month. I'd wanted to come up yesterday and today, actually, and my 9 to 5 job has me very swamped right now with deadlines, so I couldn't. So it's probably going to be, I hate to say, almost mid-October before I get back up there, but you guys definitely need to start surveys before mid-October. And all you really need to do that is going to be 3 survey sites with objectives. Why are you going to the site and what are you looking for? And Drew or Ty to go out with you guys one time. And for, you know, even if you have to split up into 2 groups, just to teach you guys how to do the basics. So I guess what I'm asking is, how are you guys looking on site selections right now? What are you thinking?
I put 3 of the coordinates in the group me that
Okay.
obviously you can take a look at, and then I offered a 4th coordinate that I sampled with a wetlands course that I think could be useful. Just getting permission from them can be a hassle sometime because it's at Nettles Park, which is, I think, the city of Central's.
Yes, that's a beautiful site. Now, I haven't, it's been a while since I've been there. Is this one intentionally planted or is the flowers that are there wild?
That's a good question. I only looked at it from a wetlands point of view, so I only looked for water. I wasn't really about any plants. And then my actual project was on birds, so we didn't care about plants either. We more so cared about trees.
Okay, okay, that's fine. And again, if you're not if you have nothing in your proposal and objectives that would um, prevent you from looking at a park on water that is pre-planted, so be it. Um, I tend to try and lead towards natural habitats because that's the way I'm trying to restore things back to. But there's nothing to say that in the future we're not having to do that anyway, right? Because as climate change causes us to lose more plants and more pollinators, we're going to have to start modifying the way we garden to try and find substitute species or other things like that. So, okay, I see your sites now, and I know where Nettles Park is. So again, your project, but what I would say is I would like for you guys to start surveying like in the next 5 days or so. I think we've already let too many weeks kind of go, and I'm not pushing it too hard, but I know the survey will continue to run throughout, at least spring and well, fall and spring through the school year for any students that decide to stick with it. But we do need to get it started because we're almost out of the traditional pollinator season. We're getting like a month and a half away. So are you guys good to do that? To just pick your 3 spots and get the permissions and coordinate times to get out there with Drew and Ty? And I'm saying this with Ty or Drew sitting on the phone, but like Drew and Ty need to be present first time because I don't have a way to just kind of meet with you all in one shot unless you guys want to choose next Wednesday to come together at the time we were originally scheduled to meet and use that hour and a half to just go outside right there in front of FEC on the campus grounds and show them how to run surveys, transects, and how to collect data. Because you could do that.
I feel like that might be better because at least for me, my schedule's crazy. So, I think it would be easier if like we could just go, like if 2 of us are free at the same time, then like we can go and do a survey, or 3 of us are free at the
Well,
same time, we can go do a survey.
well, the survey. So there's a schedule out there already. Let me pull that up. Availability. I think I put it in here. Yeah. So this right here, I'm going to share it. I used your availability, the things. Where did it go? What you guys all put in for your availability to try and create the best case scenario. There was an unfortunate circumstance where there was a couple of people that just didn't have any times that lined up with anybody else. Now, what I'm saying is if the people that want to try and meet up with them can do something outside of these times, that's fine, but just be aware that pollinator survey times are typically between 10 and 2, 10 and 3. So if you're going to choose a time, you have to pick a day. Right now, based on what you guys sent back to me, and I'm going to blow this up larger. These were the people that were able to meet. On Sundays, from 11 to 2, I had Vika, Lucinda, Vincent, and Hannah. Wednesday, 11 to 2, Caitlin, Hannah, Vika. Thursday, Drew, Ty, Kat, Vika. And that was the part that was unfortunate for me because that was like the one day both of the leaders are basically available in the same day that other people aren't necessarily there. So I think there needs to be essentially a training day with Drew and Ty. And then you guys can choose to run on this sort of rotation by picking your site. That's not saying that you have to do this every single one of these days, but I do think you guys should be able to hit up 3 sites in a week. You should be like one group should be able to go to site 1, one group should be able to go to site 2, one group to site 3. If you're sitting in a duplicate grouping, then try to group up in one of the other groups. Go ahead and use the other group, but just make sure that everyone is having access. Because like when I look at this, I see Vika was available more than really anybody, and I think Hannah had some double availability or more availability here. That's what I'm saying. Like if Hannah is available 2 days, we could potentially shift people around to try and make sure everybody's getting out there. That's the goal. Is there anyone that's sitting here with us right now that couldn't get into one of these time slots? I think Vincent had Sunday. Lucinda's here. Vika.
I'm not on there, but I could do Wednesday 11 to 2.
Kaylin?
Yes.
Okay, perfect. All right. And I feel like I'm missing someone else as well, but I've got you down now. So I just need to figure out who else. But I think it might be, did I get Nina? Nope, Nina's in there now. And Caitlin. Cool. Okay. Everybody is on this list at this point. So pick your sites, set a day with Drew and Ty where everybody can meet, and it's probably going to be better. What day were we originally meeting? Wednesday from 1 to 2:30-ish, like right in that range. I think Ty couldn't meet that day though. That was a weird one. Find a day that you all can meet with Ty and Drew. Learn how to do the surveys. You can do them right there on the hotkey lawn. Like it doesn't matter where you learn how to run the transects and how to use the devices. And you could probably even practice on some pollinators right there in the little gardens outside of Lahotsky. And then break into your teams and go out and do the surveys. And again, you don't have to do every one of these days, but you do need to do 3 surveys per week, 1 at each site. So if on week 1, this group does site 1, site 2, site 3, the following week, it might be site 1, site 2, site 3, like that, right? So that you're rotating. You guys figure out how you want to do it. Everybody good?
Sounds good.
Okay, let me know if you need anything. Sometimes I don't see the group me because it goes off with so many other activities. So if it's something very important, please message me and a regular text message or email me, either way. And I'll change the due date for this paper, and that'll give you the time to do both reformatting, adding a little bit more content, and starting to develop that methodology. Just as long as you guys are doing surveys and collecting data, we can keep working on the paper. All right, I will see you guys next week, and good luck on your quizzes, and good luck in the paper.
Thank you.
Sounds
Thank
good.
you
Thank
so
you.
much.
Thank you.
Yes. Mhm.